For much of the 20th century, genetics was viewed as a deterministic science. You were born with a fixed set of genes, and those genes were believed to govern almost everything about you, your intelligence, your risk of disease, your behavior, and even your personality. But over the past few decades, we've come to understand something profoundly liberating. The genes you inherit are not static scripts, they are dynamic instruments. And how they are played, when they are played, and how loudly they are played depends significantly on the environment you create for yourself, especially through your behaviors. This idea forms the foundation of epigenetics. Epi means above or on top of genetics. So when we talk about epigenetics, we're referring to the biological mechanisms that influence gene expression without altering the underlying DNA sequence. Think of your genome as a grand library. All the books or genes are there. But epigenetics determines which books are opened, which chapters are read, which paragraphs are emphasized, and which are skipped altogether. One of the most crucial insights from the field of epigenetics is that your daily behaviors act like signals to your genome. Every action, from what you eat, how you move when you sleep, how you think, who you interact with, and even your level of exposure to natural light, has the potential to change the expression patterns of your genes. These changes can be temporary or long, lasting, and in some cases, they can even be inherited by future generations through what we call transgenerational epigenetic inheritance. Let's explore this in more depth by looking at how this actually works in your body. At the molecular level, epigenetic changes happen primarily through three mechanisms: DNA methylation, histone modification, and non-coding RNA activity. These mechanisms influence whether genes are turned on or off, what we call gene silencing or activation. Take DNA methylation, for example. When a methyl group, a small chemical tag, attaches to a specific site on the DNA, it can silence that gene, essentially closing the book. So it's not read by the cell. Conversely, removing that methyl group can activate the gene again. These changes don't alter the sequence of the gene, but they profoundly alter its output. Over time, as you repeat certain behaviors like exercising regularly or maintaining a consistent sleep schedule, you create patterns of methylation that reinforce those behavioral effects at a molecular level. Now, what's remarkable is how responsive these epigenetic mechanisms are to lifestyle changes. For instance, in a 2008 study published in PNAS, researchers demonstrated that just 30 minutes of aerobic exercise led to measurable epigenetic changes in skeletal muscle tissue, specifically a decrease in methylation of genes involved in energy metabolism and insulin response. In other words, the body was adapting at the genetic level to become more efficient and resilient, all because of a single bout of physical activity. But the effects go even further. Let's take diet, for example. Nutrients like folate, vitamin B12, and methionine play a direct role in donating methyl groups for DNA methylation. This means that the foods you consume can literally influence the chemical architecture of your genome. For example, studies in mice have shown that genetically identical animals fed different diets during pregnancy can give birth to offspring with dramatically different coat colors, sizes, and disease susceptibilities, all due to epigenetic changes caused by maternal nutrition. It's not just about exercise and food, though. Sleep plays a massive role. Deep, restorative sleep acts like a nightly recalibration of your gene expression profile. Research from the University of Surrey found that just 1 week of insufficient sleep changed the expression of over 700 genes, many involved in immune function, inflammation, and stress regulation. In other words, poor sleep doesn't just make you tired, it alters your biological programming in ways that reduce resilience and increase vulnerability. Then there's stress, particularly chronic psychological stress. When you're under prolonged stress, your brain activates the hypothalamic, pituitary, adrenal HPA axis, flooding your system with cortisol. While acute stress can be adaptive, chronic stress has been shown to cause widespread changes in DNA methylation patterns, especially in genes related to immune function and inflammation. What's even more striking is that childhood adversity has been shown to leave epigenetic marks that persist well into adulthood, influencing mental health, immune response, and even gene expression in the brain. But here's the good news: these marks are not irreversible. Emerging research in neuroplasticity and behavioral neuroscience shows that positive behaviors can counteract or even erase many of the harmful epigenetic changes caused by stress, trauma, or lifestyle imbalance. Let's look at mindfulness and meditation. Several studies, including one from UCLA, found that consistent meditation practices can alter gene expression related to inflammation, cell aging, and neuroplasticity. Within just 8 hours of mindfulness practice, researchers found reduced expression of pro-inflammatory genes and increased activity in genes associated with antiviral responses and telomere maintenance. This brings us to a powerful concept. Behavioral epigenetics is a two-way street. Negative habits can activate genes that degrade health, but positive habits can activate genes that promote health, resilience, cognition, and even longevity. It's not a coincidence that centenarians, people who live to be over 100, often report similar lifestyle behaviors: moderate physical activity, strong social bonds, purpose, plant-rich diets, and consistent sleep routines. These aren't just surface-level observations, they reflect a deep truth. The body responds to the signals we give it, and those signals are interpreted at the genetic level. Now let's go even deeper. One of the most exciting areas of research is how social interaction affects gene expression. A landmark study at UCLA, led by Dr. Steve Cole, found that people experiencing social isolation showed increased expression of pro-inflammatory genes and decreased expression of genes involved in antiviral responses. In contrast, people with strong social support systems showed the opposite pattern. In other words, your social life is not just a psychological factor, it's a genetic influencer. Similarly, purpose and meaning in life, what we might think of as purely philosophical concerns, have measurable biological effects. People with a strong sense of purpose exhibit healthier epigenetic patterns, reduced inflammation, and better cognitive outcomes over time. This aligns with data from the Blue Zones, areas of the world with the highest concentration of centenarians, where having a purpose, ikigai, in Okinawa is a cultural cornerstone. And it's not just about your own genes. Some of the behaviors you practice today, especially during preconception or pregnancy, can influence the gene expression patterns in your children. This is what we refer to as epigenetic inheritance. For instance, animal studies have shown that male mice exposed to stress or poor diets can pass on epigenetic changes to their offspring, affecting everything from stress response to brain development. This brings us full circle to the central message. You are not at the mercy of your genetic code. You are constantly in conversation with your genome. Every action you take, every habit you form is a message, a signal that tells your genes how to behave. This is a fundamentally empowering concept. It suggests that personal health and performance are not fixed traits, but dynamic processes you can influence through informed, consistent behaviors. Yes, genetics provide the blueprint, but you are the architect deciding how that blueprint is expressed. The implication here is enormous. It means that your health is not predetermined, your brain is not hardwired, and your performance is not capped by inherited traits. Instead, you have the tools accessible, daily tools, to reshape the biological conversation happening inside your cells. And with enough time and consistency, those behaviors become encoded into your physiology, creating a kind of behavioral legacy that reinforces itself and can extend across generations. To sum this up, epigenetics doesn't promise miracles, but it does offer a scientific framework for human potential, one that's built not on magic, but on the measurable molecular outcomes of your daily choices. So whether your goal is to prevent disease, improve mental clarity, increase energy, or enhance performance, the path begins with behavior. And every moment is an opportunity to make a new epigenetic decision. When most people think about exercise, they think in terms of calories burned, weight lost, muscle gained, or cardiovascular endurance improved. These are all valid and measurable outcomes, but they barely scratch the surface of what physical activity actually does inside the body. At the molecular level, exercise is not just a mechanical action. It is a powerful genetic signal. Every time you engage in physical movement, whether it's walking, lifting weights, sprinting, or even stretching, you are triggering a complex cascade of biochemical processes that reach deep into your genome, activating specific genes and silencing others, effectively reprogramming the way your cells behave. Within minutes of beginning exercise, the body undergoes a massive shift in cellular communication. Muscles contract, heart rate increases, and blood vessels dilate to increase oxygen delivery to tissues. All of this is visible and easily tracked, but what's happening invisibly inside your cells is even more profound. As muscle fibers experience stress from movement, they release signaling molecules known as myokines. These myokines act like molecular messengers, traveling to distant tissues including the liver, brain, and fat stores, carrying with them instructions that activate genes responsible for metabolic balance, anti-inflammatory activity, and neuroplasticity. One of the first and most important genetic effects of exercise is the upregulation of genes related to mitochondrial biogenesis. Mitochondria, often referred to as the powerhouses of the cell, are essential for generating ATP, the energy currency of life. Exercise activates a transcriptional coactivator known as PGC1 alpha peroxy, some proliferator-activated receptor gamma coactivator 1-alpha, which turns on genes that lead to the formation of mitochondria. This not only improves muscular endurance but enhances energy efficiency in every tissue, from the brain to the heart. Over time, the net result is improved metabolic health, increased fatigue resistance, and greater cellular resilience against oxidative stress. Furthermore, exercise influences genes related to inflammation, which is critical because chronic inflammation is one of the major drivers of almost every age-related disease, from cardiovascular disease to neurodegeneration to certain cancers. Regular physical activity leads to the downregulation of pro-inflammatory genes and the upregulation of anti-inflammatory ones. This is largely mediated through changes in the expression of cytokines, transcription factors like NFKB, and inflammatory enzymes such as COX2. The fascinating part is that the magnitude of this effect is not entirely dependent on the intensity of exercise. Consistency plays an even bigger role. Even moderate, regular movement can powerfully shape the inflammatory gene expression profile toward a more health-promoting pattern. Another area where exercise has an extraordinary impact is in the brain. Physical activity, especially aerobic exercise, leads to the activation of genes involved in the production of brain-derived neurotrophic factor, or BDNF. BDNF is sometimes referred to as fertilizer for the brain because it supports the survival of existing neurons, encourages the growth of new neurons, and strengthens synaptic connections. In humans, elevated BDNF levels are associated with improved learning, faster memory recall, enhanced mood, and greater stress resilience. This is a genetic phenomenon not just a psychological one. Movement literally flips the switch on genes that support neuroplasticity, allowing the brain to rewire itself and adapt in meaningful ways. In fact, the genetic effects of exercise on the brain are so significant that some researchers now propose exercise as a frontline treatment for depression and cognitive decline. In the context of aging, exercise modifies the expression of genes involved in cellular repair and longevity. A particularly fascinating target of exercise-induced gene regulation is the family of sirtuins. Sirtuins are a group of proteins that help regulate cellular health and are closely tied to aging and metabolic efficiency. They depend on the availability of NAD plus nicotinamide adenine dinucleotide, which also increases with exercise. These proteins play a role in DNA repair, stress resistance, and mitochondrial function. Sirtuin gene expression is upregulated following consistent physical activity, suggesting that exercise acts as a molecular defense system against aging. Another vital mechanism through which exercise affects gene expression is epigenetics specifically, changes in DNA methylation patterns. These are chemical tags that turn genes on or off. A 2012 study published in the journal Cell Metabolism showed that even a single session of exercise led to immediate changes in the methylation of genes in human skeletal muscle, particularly genes involved in energy metabolism and insulin sensitivity. What's important here is that these changes are both rapid and reversible, which implies that our genomes are highly responsive to movement. Over time, repeated bouts of exercise create a kind of epigenetic memory in the muscle tissue, making those genes more responsive to future activity. This is part of why physically active people adapt to stressors more efficiently and recover faster. When it comes to insulin regulation and glucose metabolism, the gene-level effects of exercise are equally striking. In both diabetic and non-diabetic individuals, physical activity increases the expression of GLUT4, a glucose transporter that allows muscles to absorb glucose from the blood. The more active you are, the more GLUT4 is expressed, and the more efficiently your muscles can clear glucose, reducing the burden on the pancreas and stabilizing blood sugar levels. This isn't just about burning off sugar, it's about reprogramming your body's ability to handle it. Exercise also has hormonal effects that are partially mediated through changes in gene expression. For example, physical activity increases the expression of genes responsible for testosterone synthesis in men and estrogen balance in women. It also influences genes that control the sensitivity of tissues to hormones like insulin, leptin, and cortisol. This hormonal, genetic interplay is one of the key reasons why physically active individuals tend to have more balanced endocrine profiles, greater stress tolerance, and fewer metabolic disruptions. We also see exercise influence the immune system in fascinating ways. Beyond reducing systemic inflammation, regular physical activity upregulates genes involved in immune surveillance and response. For instance, natural killer cells, key players in identifying and destroying virus. Infected and cancerous cells become more active and more abundant as a result of consistent exercise. The expression of genes responsible for immunoglobulin production and lymphocyte function is also enhanced. What this means is that physically active people are not just healthier, they're more immunologically vigilant. Emerging research also suggests that exercise-induced gene expression has profound effects on the gut microbiome. While the microbiome itself doesn't contain human genes, it has a dynamic interplay with the host genome. Physical activity increases the diversity of gut microbes and enhances the expression of host genes involved in intestinal barrier function, short-chain fatty acid metabolism, and immune tolerance. This gut genome communication loop appears to be crucial for maintaining whole body health and is one of the reasons why exercise has systemic effects that extend far beyond the muscles. Even reproductive health is influenced at the gene expression level by exercise. In men, physical activity improves sperm quality through epigenetic modifications, which may have implications not just for fertility, but for the health of future offspring. In women, regular movement helps regulate ovulatory cycles and modulates gene expression in the endometrium, improving reproductive efficiency. These changes are not just hormonal, they are deeply rooted in gene regulation pathways. One of the lesser, known but highly important areas where exercise exerts genetic influence is sleep. Physical activity during the day leads to the expression of genes that enhance sleep quality and deepen slow-wave sleep. For example, genes that regulate circadian rhythm known as clock genes are modulated by physical movement. These include clock, BMAL1 and PER genes, which govern sleep-wake cycles, hormone release, and metabolism. Regular physical activity helps synchronize these genes with environmental cues like light and dark, leading to improved circadian alignment and restorative sleep. And if we bring this conversation into the world of mental health, we find that exercise influences genes involved in serotonin signaling, dopamine transmission, and endorphin regulation. This is part of the reason why physically active people tend to experience lower rates of depression and anxiety. The expression of COMT, MAOA, and tryptophan hydroxylase genes is modulated by physical activity in ways that enhance mood and emotional resilience. Again, these are not simply psychological effects, these are molecular changes that affect how neurotransmitters are synthesized, released, and recycled. The type of exercise you do also matters in terms of the specific genetic effects. Resistance training primarily influences gene expression in muscle hypertrophy pathways like the mTOR signaling cascade, leading to increased protein synthesis and muscle fiber remodeling. Endurance training, on the other hand, leans more heavily into mitochondrial gene networks, vascular adaptation, and oxidative metabolism. High-intensity interval training it seems to provide a unique hybrid effect, activating both anaerobic and aerobic gene programs, making it especially potent for time-efficient cellular reprogramming. It's worth noting that the cumulative genetic effects of exercise are not only preventive, they are also corrective. In people with existing chronic diseases like obesity, hypertension, or metabolic syndrome, exercise has been shown to reverse maladaptive gene expression patterns. For example, in type 2 diabetics, consistent exercise reduces the expression of genes associated with insulin resistance while increasing the activity of those involved in glucose transport and lipid metabolism. In patients with cardiovascular disease, exercise enhances the expression of endothelial nitric oxide synthase (eNOS), improving vascular function and reducing the likelihood of further plaque buildup. What this all points to is a central truth in biology. Exercise is a molecular switch. It is one of the few interventions that affects virtually every organ system and does so by altering gene expression in a precise and beneficial manner. It operates not just at the level of muscles and joints, but at the level of chromosomes and histones. And importantly, the genome remembers, with consistent effort, the body retains epigenetic marks that make future adaptation easier, recovery faster, and performance higher. These gene expression changes are not instantaneous, but they are rapid enough to matter and stable enough to build upon. That means each workout, each bout of movement, is not just about improving how you look or feel in the moment. It is an investment in the way your genes will behave days, weeks, and even years from now. It is biology in action, plastic and responsive, and it's entirely within your control. Each night, when you lie down and fall asleep, you may think of it as a time when your body simply shuts down to rest. But biologically, that couldn't be further from the truth. Sleep is one of the most active and dynamic periods in your 24-hour physiological cycle. While your conscious awareness disappears, a sophisticated array of molecular and cellular processes is launched, processes that reach deep into your genome. Your genes, thousands of them, are either turned on or off based on whether you're asleep, how deeply you're sleeping, and when that sleep occurs. In other words, sleep is not just restorative, it is instructive. It tells your genes how to behave, which in turn shapes your health, performance, immune system, and even your aging process. Research in chronobiology and molecular genetics has shown that gene expression is profoundly rhythmic. Certain genes are expressed only during the night, others only during the day. This cyclical nature of gene activation is what we refer to as circadian regulation, and it's governed by molecular clock genes such as clock, BMAL1, PER, and CRY. These genes create feedback loops that operate on a 24-hour cycle and regulate not only your sleep, wake patterns, but also metabolism, hormone release, body temperature, cognitive performance, and immune response. When you sleep at the right time and in the right depth and duration, you synchronize this entire system. But when your sleep is insufficient, irregular, or mistimed, such as during shift work, jet lag, or chronic sleep restriction, those same genes become desynchronized. The downstream result is a chaotic cellular environment in which tissues, organs, and even immune cells operate out of sync, leading to dysfunction. For instance, during deep, slow-wave sleep, what we call non-REM sleep, genes associated with cellular repair and detoxification become highly expressed. These include genes responsible for DNA repair, protein folding, and the removal of damaged mitochondria. One critical example is the upregulation of genes involved in the production of antioxidant enzymes like superoxide dismutase, SOD, and glutathione peroxidase. These enzymes neutralize reactive oxygen species Rhos, protecting cellular structures and maintaining mitochondrial health. When sleep is shortened or fragmented, the expression of these protective genes diminishes, allowing oxidative stress to accumulate. Over time, this contributes to neurodegeneration, cardiovascular issues, and accelerated aging. Another compelling area where sleep and gene expression intersect is immune function. During high-quality sleep, the body ramps up the expression of genes that control the activity and sensitivity of T cells, B cells, and natural killer cells. These are the frontline defenders of your immune system. Genes responsible for the synthesis of cytokines such as interleukin-2 and interferon gamma become active, preparing your immune system to identify and eliminate pathogens. Simultaneously, sleep downregulates genes involved in the expression of pro-inflammatory cytokines like interleukin-6 and TNF-alpha. This balance between immune readiness and inflammatory suppression is critical. When sleep is disrupted, we see the exact opposite: inflammation. Related genes are overexpressed and immune surveillance genes are suppressed. This imbalance makes individuals more susceptible to infections, slows recovery, and even decreases the efficacy of vaccines, a phenomenon documented repeatedly in sleep deprivation studies. Cognitive performance is another dimension profoundly shaped by the gene regulating power of sleep. The brain uses sleep as a time to consolidate memories, regulate emotional responses, and restore synaptic plasticity. Genes involved in long-term potentiation, LTP, the strengthening of synapses that underlies learning, are selectively expressed during REM sleep and deep sleep. For example, genes regulating the expression of BDNF brain-derived neurotrophic factor peak in certain stages of sleep and drop precipitously in response to sleep deprivation. Since BDNF is vital for the formation of new neural connections, its suppression means impaired learning, reduced problem-solving ability, and even diminished creativity. Students, athletes, and high performers who skip sleep in favor of extra work or training may be undermining the very neural adaptations they're trying to achieve simply by disrupting expression of the genes that make those adaptations possible. Even short-term sleep deprivation has been shown to alter the expression of over 700 genes involved in metabolism, immune function, and circadian regulation. In a pivotal study published in the Proceedings of the National Academy of Sciences, researchers found that a single week of sleeping fewer than 6 hours per night was sufficient to dysregulate gene expression patterns across the body. Among the most affected were genes involved in inflammatory responses, stress regulation, and lipid metabolism. These findings suggest that even short-lived disturbances in sleep can produce widespread and measurable changes in the genomic architecture of health. The endocrine system, especially hormones like cortisol, testosterone, and growth hormone, is deeply intertwined with sleep-mediated gene expression. Cortisol, your body's primary stress hormone, is governed by the HPA axis and follows a circadian pattern. Its release is influenced by genes like CRH and ACTH, which are tuned by the quality and timing of your sleep. Under normal conditions, cortisol levels rise just before waking, giving you energy to start the day and taper off at night. However, when sleep is erratic or inadequate, the expression of these regulatory genes becomes disordered. Cortisol remains elevated at night, impairing deep sleep, and fails to rise appropriately in the morning, leading to fatigue and poor cognitive performance. Long-term misalignment of this rhythm contributes to insulin resistance, fat gain, and even mood disorders. Testosterone and growth hormone, both of which play critical roles in tissue repair, muscle growth, libido, and cognitive function, are secreted primarily during deep sleep. This secretion is genetically orchestrated by the timed expression of genes like GH1 for growth hormone and LHCGR for testosterone synthesis. When deep sleep is compromised, the genetic signals that initiate and sustain their release are also blunted. For athletes or anyone engaged in resistance training or recovery, heavy protocols, this reduction has direct implications on performance and adaptation. Simply put, if you aren't sleeping properly, your body isn't accessing the gene programs that build muscle or restore hormonal equilibrium. Melatonin, often thought of as merely a sleep hormone, is also an epigenetic regulator. It influences gene expression in the brain, immune system, and even in cancer pathways. Melatonin receptors are found in nearly every tissue of the body, and melatonin itself regulates clock genes, suppresses oncogenes, and enhances the expression of tumor suppressor genes. However, melatonin in synthesis is profoundly sensitive to light exposure, particularly blue light. Exposure to screens or artificial lighting late into the evening suppresses melatonin in release and disrupts its gene modulating actions. This is one of the reasons why sleep hygiene practices like dimming lights, reducing screen time, or using blue light blockers can have measurable biological effects. They protect circadian, controlled gene networks responsible for long-term health. Sleep also affects metabolic genes, particularly those regulating appetite, fat storage, and glucose utilization. Two of the most studied hormones in this context are leptin and G-renin. Leptin, which signals fullness, and G-renin, which stimulates hunger, are both regulated by genes whose expression is modulated by sleep. Sleep deprivation shifts this balance, increasing G-renin ghrelin and decreasing leptin, thereby encouraging overeating and fat gain. At the gene level, we also see a reduction in insulin receptor sensitivity and a downregulation of genes involved in lipid oxidation, such as CPT1 and PPAR-alpha. This helps explain why even a few nights of poor sleep can impair glucose metabolism and increase fat accumulation, an effect that, when repeated over time, elevates the risk for obesity and type 2 diabetes. And then there is the link between sleep and longevity. Telomeres, the protective caps on the ends of chromosomes, shorten as we age. Shortened telomeres are a biomarker of cellular aging and are linked to an increased risk of chronic disease and early death. Intriguingly, sleep quality has been shown to affect the expression of genes involved in telomerase activity, the enzyme that maintains telomere length. In people with chronic insomnia or sleep apnea, we see reduced telomerase activity and accelerated biological aging. Conversely, individuals with high sleep efficiency and good sleep timing show more favorable expression of longevity, associated genes, and slower telomere attrition. Another key aspect is that sleep is not a uniform state. It is composed of cycles roughly 90 minutes each that include stages of light sleep, deep sleep, and REM sleep. Each of these stages has a unique molecular signature with different sets of genes expressed depending on the phase. Deep sleep favors the expression of genes involved in physical repair and immune readiness, while REM sleep enhances neuroplasticity and emotional memory integration. Fragmenting these cycles through alarms, caffeine, alcohol, or blue light disrupts the precision timing of gene expression and diminishes the benefits of sleep, even if total sleep time remains the same. This is why sleep quality often trumps quantity, and why improving the continuity of sleep is a direct way to protect gene-regulated processes. An often overlooked but crucial factor is the timing of sleep in relation to your circadian rhythm. The body's internal clock is genetically encoded but can be shifted by behaviors such as light exposure, meal timing, and social cues. Going to sleep outside of your biological night, say, sleeping from 3 a.m. to 11 a.m. instead of 11 p.m. to 7 a.m., even if you get 8 hours, results in a misalignment between external behavior and internal gene regulation. This condition, known as social jet lag, disrupts the synchronization of peripheral clocks in organs like the liver, pancreas, and gut, leading to a breakdown in coordination among tissues. Over time, this dyssynchrony has been shown to increase the risk for cardiovascular disease, cancer, and mood disorders. Ultimately, what we're seeing is that sleep is not just a passive rest state. It is a master regulator of gene expression across virtually every system in the body. It orchestrates molecular rhythms that support immunity, cognitive performance, tissue repair, metabolic health, and aging. Every night, your behavior around sleep either promotes or hinders these genomic processes. And what's most empowering is that many of these effects are reversible. Improving your sleep quality by optimizing light exposure, maintaining consistent sleep times, and minimizing sleep disruptors can recalibrate gene expression patterns in your favor, often within days to weeks. Sleep is not just about feeling rested. It's about accessing the full capacity of your genetic blueprint to perform, heal, defend, and thrive. Your genome is listening while you sleep, responding to the signals you send it with your habits, environment, and daily rhythms. In that sense, sleep is one of the most powerful forms of epigenetic therapy available to us, and it's free, biologically ancient, and available every night. When the body is exposed to cold, it interprets this not as a threat, but as a significant environmental challenge that demands a coordinated response at every level: molecular, cellular, systemic, and behavioral. What emerges from this challenge is not just a feeling of alertness or the sense of having overcome discomfort, but a cascade of biological changes that alter gene expression in ways that enhance resilience, regulate metabolism, reduce inflammation, and promote mental clarity. The more we study the effects of deliberate cold exposure, the more we understand that it's not just a temporary shock to the system, it's a form of environmental hormesis, a beneficial stressor that recalibrates multiple biological pathways for improved long-term performance and health. At the genetic level, one of the first changes triggered by cold exposure involves the activation of cold shock proteins, particularly RNA-binding motif protein 3 RBM3 and cold inducible RNA-binding protein SERP. These proteins are upregulated in response to a drop in body temperature and play key roles in stabilizing and preserving mRNA transcripts within cells. In simpler terms, they help maintain the integrity of gene expression during environmental stress. This is critical in neurons, where mRNA stability directly supports neuroplasticity, the brain's ability to adapt and form new connections. Studies have shown that RBM3 expression correlates with the preservation of synapses during cold stress and even contributes to cognitive resilience in neurodegenerative conditions. In animal models, upregulation of RBM3 is associated with the regeneration of lost neural connections and protection against synaptic degeneration. This alone provides a powerful rationale for why cold exposure may have cognitive and neuroprotective benefits when practiced safely and consistently. But the effects of cold go well beyond preserving neurons. One of the most studied pathways involves the sympathetic nervous system. Upon cold exposure, norepinephrine is released in large quantities from sympathetic nerve endings and from the adrenal medulla. This hormone, which functions as both a neurotransmitter and a signaling molecule, spikes within minutes of entering cold water and remains elevated for up to several hours afterward. Norepinephrine binds to adrenergic receptors throughout the body and brain, producing effects ranging from increased attention and vigilance to enhanced immune readiness. On a genetic level, it activates a set of transcription factors that increase the expression of anti-inflammatory cytokines such as interleukin-10 and decrease the expression of pro-inflammatory genes like TNF-alpha and IL-1 beta. This rebalancing of immune gene expression may be one of the reasons why people who regularly engage in cold exposure report fewer colds and reduced inflammation-related symptoms over time. Another remarkable effect of cold is its impact on adipose tissue, particularly the conversion of white fat into metabolically active brown and beige fat. Brown adipose tissue BAT contains a high number of mitochondria that express a gene called UCP1 uncoupling protein 1. This protein allows the mitochondria to generate heat instead of ATP, a process known as non-shivering thermogenesis. Cold exposure, especially when repeated over days or weeks, upregulates the expression of UCP1 and other thermogenic genes like PGC1 alpha and PRDM16 in adipocytes. These genes don't just respond to cold, they transform fat cells into calorie-burning heat engines. This has profound implications for metabolic health. Individuals with higher BAT activity have improved insulin sensitivity, lower blood glucose levels, and reduced visceral fat, all of which are correlated with lower risks of metabolic syndrome and cardiovascular disease. Interestingly, cold exposure doesn't just influence fat metabolism, it also affects muscular and cardiovascular gene networks. The shock of cold activates AMP-activated protein kinase, a cellular energy sensor that responds to changes in ATP levels. AMP in turn upregulates genes involved in mitochondrial biogenesis, fatty acid oxidation, and glucose uptake. This is the same pathway activated by exercise and fasting, and its activation through cold represents yet another way the body adapts to stress by becoming more metabolically efficient. This makes cold exposure an effective adjunct to exercise for those looking to enhance endurance, improve energy utilization, and stimulate adaptive cellular stress responses. For example, genes like PPAR, delta and SIRT1, which are involved in endurance adaptation and cellular longevity, are activated through repeated cold exposure. From the perspective of mental performance, cold stimulates the hypothalamic, pituitary, adrenal HPA axis, leading to transient increases in cortisol. While chronic cortisol elevation can be harmful, brief spikes, especially those timed early in the day, can enhance alertness, mental clarity, and readiness. This aligns with the psychological phenomenon often reported by cold practitioners, a sharpening of mental focus and a reduction in anxiety. Part of this effect may be mediated by changes in the dopamine system. Cold exposure, especially to water below 15 degrees C, 59 degrees F, has been shown to increase circulating dopamine levels by up to 250%, a powerful effect comparable to pharmacological agents. Dopamine doesn't just regulate motivation and mood, it also influences goal-directed behavior and executive function by modulating the prefrontal cortex. At the genetic level, this is supported by cold-induced upregulation of dopamine receptor D2, DRD2 mRNA expression, suggesting that cold exposure may sensitize the brain to dopamine signals, making you more focused, driven, and resilient. The cardiovascular benefits of cold are equally compelling. When you immerse yourself in cold water, blood vessels constrict to preserve core body temperature, a process known as vasoconstriction. After leaving the cold, the vessels dilate again, a rebound effect that enhances circulation. Over time, this cycling of vasoconstriction and vasodilation improves vascular tone and endothelial function. Studies show that repeated cold exposure can increase nitric oxide availability by upregulating endothelial nitric oxide synthase (eNOS) gene expression. Nitric oxide is a potent vasodilator that reduces blood pressure and enhances blood flow to muscles and organs. Improved endothelial function is strongly associated with reduced risk of cardiovascular events, better exercise capacity, and longer lifespan. There's also a resilience building effect, both physiological and psychological, that comes from deliberately exposing yourself to discomfort. This ties into the concept of stress inoculation, the idea that voluntary exposure to controlled stress enhances your capacity to handle future stress. The physiological basis of this adaptation involves hormetic gene networks, genes that are upregulated in response to stress but confer long-term protection. Examples include FOXO3, a transcription factor associated with longevity and cellular repair, and heat shock proteins, yes. These are also activated by cold, which help refold damaged proteins and prevent cellular stress. Over time, repeated cold exposure trains these systems to respond more efficiently to future insults, be it psychological stress, physical exertion, or environmental changes. One of the most profound long-term effects of cold exposure is its impact on inflammation, which is increasingly recognized as a root cause of many chronic diseases. Cold activates the vagus nerve, a major component of the parasympathetic nervous system, which in turn suppresses inflammatory gene pathways through what's known as the cholinergic anti-inflammatory reflex. Through this mechanism, the brain can directly modulate the immune system via the acetylcholine-mediated inhibition of pro-inflammatory cytokines. This is not speculative. Studies on cold water immersion have shown reductions in markers like CRP, C-reactive protein, and improvements in conditions such as rheumatoid arthritis, asthma, and inflammatory bowel disease. The genetic underpinning involves the downregulation of NF-kappa B, a master regulator of inflammatory gene expression. This transcription factor is normally activated by stress and infection, but remains quiescent in individuals who regularly expose themselves to brief, controlled cold stress. Cold exposure may even play a role in promoting longevity at the cellular level. In model organisms such as C. elegans and mice, exposure to colder environments extends lifespan, an effect mediated by genetic pathways involving insulin signaling and oxidative stress response. In humans, we see indirect parallels. Lower core body temperatures correlate with reduced metabolic rate and longer lifespan. At the gene level, cold exposure has been shown to enhance the expression of sirtuins, particularly SIRT3, which regulates mitochondrial efficiency and DNA repair. These longevity genes are normally activated during calorie restriction and exercise, but appear to respond similarly to cold. Of course, it's important to recognize that the benefits of cold are highly dependent on the dose and context. Too much cold, too often, or without proper safety can lead to adverse effects like suppressed immune function, chronic fatigue, or even hypothermia. The key is in creating short, deliberate, and progressively challenging exposures. Most research and anecdotal reports converge on the idea that 11 to 15 minutes per week, broken into multiple sessions of 2 5 minutes at a time, provides substantial benefits when using water temperatures below 15 degrees C, 59 degrees F. These exposures are enough to engage the pathways described without overwhelming the system. What makes cold exposure such a powerful behavioral tool is its immediacy. Unlike other interventions, such as exercise or nutrition, which often take weeks to months to manifest effects, cold produces a noticeable shift in state almost instantly. The elevated norepinephrine, increased dopamine, the sense of mental clarity, the improvement in breathing patterns, all of these emerge within minutes of exposure. But beneath that quick state change is a deeper, slower process, the epigenetic remodeling of your physiology. Over time, the genes responsible for heat production, inflammation control, metabolic efficiency, and neural resilience become more active, leading to improved performance not just in the cold, but in all aspects of life. To put it plainly, cold exposure gives you access to your biology's reserve systems. It recruits mechanisms that are usually dormant under comfortable conditions. These include not only molecular systems like cold, shock proteins, and thermogenic genes, but also neurochemical circuits involved in mood, attention, and adaptation. With consistent practice, this behavioral tool reshapes the molecular landscape of your body, teaching it to be more efficient, more resilient, and more responsive to life's demands. And importantly, cold exposure reinforces agency. When you choose to step into discomfort, when you activate your sympathetic nervous system deliberately and then calm it intentionally through breath or mental focus, you are building not only biological resilience but psychological fortitude. The effect is dual. The body becomes more adaptive and the mind becomes more disciplined. That duality is rare in most interventions and is part of what makes cold exposure such a powerful enhancer of human potential. Thank you for your time and attention. What we've explored today is not just a physiological tool, but a powerful intersection between behavior and biology. Deliberate cold exposure, when practiced safely and consistently, doesn't just change how we feel, it changes who we are at the cellular and genetic level. It teaches the body to become more efficient, the mind to become more resilient, and the entire system to adapt in ways that optimize health and performance. Remember, discomfort, when chosen deliberately, can be a doorway to growth. Use cold as a catalyst, not just for physical enhancement, but as a practice in discipline and mental mastery. The genes you express today are shaped by the behaviors you engage in consistently. Be strategic, be safe, and above all, be consistent. Thank you again for being here and for your commitment to learning and improving. Wishing you clarity, strength, and resilience always. Stay curious and stay cold.